Everyone to our brain tumor webinar series. My name is Christy Allen and I am a board member with BTFC, and I'm really excited to welcome our guest today, Dr. Yan Wan, to talk about developing artificial intelligence tools to monitor brain metastases in the population. We'll start with a land acknowledgement. So we acknowledge that the Brain Tumor Foundation of Canada is located on the traditional territory of the Anishinaabe, Haudenosaunee, Attawandaron, and Wendat peoples. We honor and respect the history, languages, and culture of the diverse Indigenous people who call this territory home. We move forward with a profound sense of gratitude and respect for the land and its original caretakers, and are grateful to have the opportunity to work and live in this territory. So welcome to our webinar series. This series is designed for anyone impacted by a brain tumor, patients, survivors, caregivers, and volunteers, as well as healthcare professionals and researchers seeking to expand their knowledge. So our sessions will explore a wide range of topics, including neuro-oncology, neurosurgery, research advancements, psychosocial oncology, mental health, and caregiver support. So a couple housekeeping notes. The webinar will be recorded and a link to the recording will be shared with all registrants within 1 week of the webinar, and the recording will also be available on our website. We do have dedicated time at the end of the presentations for questions and discussions, so please send your questions through the Q&A function. And take note that we won't be able to answer individual specific questions about specific circumstances, but we encourage you to contact your health team to get specific advice. And you can also reach out in the chat to connect with the speaker and the panelists. So now we'll move into some quick poll questions just to learn a bit more about who's in the audience today. Wonderful. Thank you so much for participating in our polls. So I'm very excited to introduce today's speaker. Dr. Wan is a professor at the University of Alberta School of Public Health and received an undergraduate degree from the Nanjing University in China in 1996, followed by an MSc from Michigan State University and a Master of Mathematics in Biostatistics and a PhD in Statistics, both from the University of Waterloo. Dr. Wan's academic career focuses on cancer research. She led and collaborated in projects ranging from prevention, surveillance, diagnostics and screening, health service and survivorship with over 70 publications, of which 15 are on the brain tumor surveillance in Canada. A recipient of the Early Career Award from the Canadian Institute of Cancer Research in 2017, Dr. Wan has received major research grants, including from the Canadian Cancer Society, Canadian Institutes of Health Research, Natural Sciences and Engineering Research Council of Canada, and National Institute of Health in the US. So welcome, Dr. Wan.
Thank you. Thank you very much, Christy, for your kind introduction. And thank you. Good afternoon, everyone. Thank you for taking the time from your busy schedule to come to my talk today. I'm just go ahead to share my screen. Let me do that. They're looking right now for everyone?
Yep, looks good.
Okay, thank you. So it's a great pleasure to present our work at the Brain Tumor Foundation of Canada's webinar series. We know the Brain Tumor Foundation of Canada has been a steadfast supporter of our research group, the Brain Tumor Surveillance Research Collaborative of Canada. I'm going to focus my talk on our recent project, which is titled here, Developing Artificial Intelligence Tools to Monitor Brain Metastasis in the Population. But before I go into this topic, I'd like to briefly talk about of the BTRSC, including its origin and overall objectives. So the Brain Tumor Surveillance Research Collaborative of Canada, its predecessors is Brain Tumor Registry of Canada. In fact, our website still carry this name. Our group conceptualized in 2016 and is established in 2019. It was established to address the lack of surveillance data of central nervous system tumors in Canada, commonly known as brain tumor. Prior to our work, Brain Tumor Registry of Canada, to this work, the neuro-oncology community in Canada's default is to use USA's data, which is from Central Brain Tumor Registry of the United States, CB Trust. They use their CNS tumor estimates and extrapolate them onto the Canadian population, which is far from ideal. So our goals at BTSRC is to improve the infrastructure for identifying and reporting on all primary CNS tumors in Canada, to capturing, as you probably all know, the brain tumor had a high diversity. We want to capture this diversity of these tumors. While it only accounts for like 2% of all cancers in Canada, in Canadian population, Canadian cancer population, it's actually had a huge variety in it. So we want to make sure that information is being captured. And we want to assessing and improving the quality of the surveillance data currently we have on these tumors. We provide the biannual high-quality surveillance estimates, including incidence, prevalence, and survival rates. Other than this routine work we do, we also conduct relevant surveillance researches on these brain tumors to provide accurate information to policymakers and health providers. So we use the Canadian Cancer Registry data. So the Canadian Cancer Registry, essentially, it's a collection of all provincial cancer registry data. So all the province under their own province legislation, they have, their data collecting cancer surveillance role, and they collect their data within their province, and then they submit the data to Statistics Canada, and then Statistics Canada have this cancer division that will scrutiny all these data together and pull them together and create this thing called Canadian Cancer Registry data, which is available to research use through research data center. Well, So I'm going to briefly just touch on the two projects we have done with this CCR data. One is looking at the data quality for CNS tumors in CCR data. The other is using CCR data to give us an idea, an understanding of the prevalence of the CNS tumors. So this is what we did for the 2013 to 2018 CCR data on CNS tumors. We assessed the CCR data quality. So on the x-axis, on this figure, on the x-axis, we have this thing called age standardized incident rate per 100,000 person year. And we have to see different two colors. One is the United States data based from the CB Trust, and the other is from Canada. The orange color is from Canada, based on the CCR data. And these, because cancer is a very age-dependent disease. So in order to compare across different populations, we need to do this age standardization. So that's the idea behind that. And what we also stratified by malignant CNS tumor and non-malignant CNS tumors. As we can observe from this graph, the United States had a much higher non-malignant CNS tumor incident rate comparing to Canadian. And this is really speak to the fact of the under capture of non-malignant CNS tumors in the Canadian cancer registry systems. So that's one thing we identified. But in terms of malignant tumor, as we can see from the graph, both United States and Canada have a similar age standardized incident rate, like Canadian being slightly higher. Another thing we identified, as I explained, all this registry data has been submitted by each provinces. We can actually look at each provinces individually. We found that in Ontario's non-malignant CNS tumor incidence rate is the closest to the expected, or let's say the US rate, which is the Ontario had a 16.56. Comparing to the US is close to 17-ish here. And however, within the non-malignant tumor in the Ontario data, 6.43 out of these 16 are unclassified tumors. And this speaks to the, again, speak to the data quality issues in our own registry. Okay, so the other project we looked at, one thing we looked at is the prevalence of the brain tumors. So prevalence is basically saying at a specific time point, in this case, January 1st, 2018, how many people is living and diagnosed with this particular condition. And what we show here in this particular graph is we separated by malignant CNS tumor and non-malignant CNS tumors. So for malignant CNS tumors, again, we stratified by male and female, male in blue and female in orange color here. We found that there's more male patient lived with malignant CNS tumors, 4,170 versus female is 3,190. And this graph here showed the top 4 malignant CNS tumor by counts, by frequency, by prevalence here. Essentially, they are from GBM, polycystic astratoma, oligodendronoma, and glioma malignant NOS. And in the other hand, for the non-malignant CNS tumors, we found more female patients live with non-malignant CNS tumors. For the male, there's 12,185. For the female, it is 18,385. And it's dominated by meningioma, followed by unclassified tumors, and the third one is a tumor of cellular region. Again, I'm only pick the top three to display because there's, as you may know, a lot of different brain tumors. So for most CNS tumor groups, we see that there's more male patient living with this disease than female patients. Okay, so this is just like a brief snapshot of some things we have done by our BT-SRC group. I'm going to go into my main talk right now, and this is actually the first time I present this work to any audience. So please forgive me if I'm being not very smooth and not very polished yet. So as we know, we may know, the brain metastasis is a serious complication in cancer patients. Let's see, they cause debilitating neurological symptoms. They reduce quality of life and also impact survival. However, the incidence rate of the brain metastasis, which is also called a secondary brain tumor, their incidence rate is not, there's a lack of knowledge basically there. There are, from different population studies, there's an incidence rate of the BM ranging from 7 to 14 persons per 100,000 person year across different population studies. And there is a hypothesis that the incidence of the BM, I'm going to use BM to short phrase for the rest of the talk, is thought to be increasing, mainly due to the improved survival of patients owing to the treatment of their primary cancers being improving and more treatments. And also as the improved detection of cancers due to the wider introduction of advancing neuroimaging diagnostic technologies. So using the estimates from published population-based study and the Canadian Cancer Registry, we estimated there's about 10,000 Canadians are diagnosed with malignant brain tumors annually, of which 8,000, like that is 80%, are these BMs. But however, there's only about 2,800 of them are being captured by the Canadian Cancer Registry, because a Canadian Cancer Registry, they will capture the brain mets when they were diagnosed at their cancer diagnosis. So what it means is if some patient developed the brain mets during their follow-up years, during the treatment, during the years after treatment, these information are not being captured. So there is really a knowledge gap there. There's a lack of information on when, how, often, and in whom these BM occur. And this lack of this knowledge gap is very well recognized. The challenge being, how do you capture those data? How do you monitor and how do you get this data? This is very difficult because it requires a manual search of every single cancer patient's record on a routine basis by highly qualified cancer registrars, which is a real substantial barrier to obtain this very important data. At the same time, we do recognize the importance of this data because increasing capture of this information would substantially increase the efficiency of the health service planning and research, and helps develop treatment strategies resulting in improvement in outcomes. So we were thinking about to address this challenge. So, and as we indicated, the brain mets, a lot of time they are captured due to this advanced imaging technology. And this is a sample of radiology report, which shows you what a radiology report is, and it's on the head, MRI report. It contains a history, why the patient is here for this study, whether or not there's comparison of prior imaging study from the same patient for the same organ, what kind of techniques being used, what are the findings, which is a very detailed observation that the radiologist describe from the imaging study. And then lastly, there's this impression session, which gives an overall assessment of whether radiologist diagnosis, basically, and also follow-up recommendations. So we think, okay, we might be able to use an artificial intelligence approach to capture this information. And we proposed that idea to develop a natural language processing NLP algorithm utilizing specific large language models to capture this BM diagnosis in those radiology reports. We kind of have conceived the idea back in 2017. We published it in 2018, and we got a grant, I think, in 2020, 2021 to start this work. So before I go into the details, I'm just trying to get out of what is a large language model and what is NLP. And these are based on the artificial intelligence overview from Google. So large language model is simply a sophisticated artificial intelligence technology that excels at understanding and generating human-like text. It is trained on vast amount of text data, enable it to grasp the nuances of the language and produce meaningful, relevant response. They are used in various applications like chatbots, translation, text generation these days. I think most people must have heard about it. And in terms of what is natural language processing, it is a broader field of artificial intelligence focus on enable the computers to understand and interact with human language. In essence, NLP is umbrella, and the large language model, is a specialized tool used within that umbrella. Okay, so our approach, we've built a study cohort, which are patients who diagnosed with cancer between 2012 and 2020, with follow-up time until 2022, as captured by the Alberta Cancer Registry. So basically, we use Alberta Cancer Registry to identify cancer patients, okay, between this specific time frame. And for the data, we find for this study cohort, we pulled all the radiology reports of head CT or the MRI scan of the brain between that time frame, 2012 to 2022, from the Alberta's centralized radiology reports database. And they were linked to the patient cancer diagnosis information in Alberta Cancer Registry. So in total, there's 112,000 cancer patients in that time frame, and there is a 300,000 radiology reports put on these patients. Of course, majority of them, yes, more than half of them will not have any head scan, but some of them would have a lot of head scans as well. So one thing, advantages actually, I want to point out, we can do that in Alberta because Alberta have this centralized radiology reports database. This is not true for British Columbia and Ontario. So we do have this one centralized database that has everybody's head imaging information, radiology reports. Again, we see that we have seen that already. So I just want to using this slide to say that we use this model. We use this large language model called BioBERT. It was trained on medical text, clinical text, So it has a good understanding of medical terminology. For example, this word, I don't understand myself, right? I'm not a doctor, a physician myself, so it will be very hard for a layman to understand. But this BioBERT large language model has been trained on this clinical database, so they kind of understand this language much better than I do, at least. And so we used this large language model as a base model to train and use our data to develop using the NLP approach to develop our model, if it makes sense. And the other thing is, because we use the BioBERT model, the BioBERT model only takes 512 words. It can only process 512 words. Sometimes the whole report is going to be longer than 512 words. So in order to deal with that situation, we actually we actually only used the finding session and the impression sessions from the radiology reports, because those are the place where the information are most rich for, for the relevant, whether or not there is brain mets there. And that gives me this is the overview of how we developed our model. So essentially, we start with a CT, MRI scan, basically the reports, right? It's starting from the scan of the brain and head, and the radiologist generate a text report. We had a annotator. We had a annotator who read this report and label this report to be BM yes and BM no. Okay, so this information, yes and no, these are important labels. They were used to train our model. From the text report, we have to do first some data pre-processing to remove some words that are wrong there, because it's converted from text to PDF, so there's a lot of technical details I'm going to just omit here. And we have split the data into training data and testing data. So training data is used to train the model, tested data is used to evaluate the model performance. And as I explained, the training data, we segment it into these two sessions, finding session and impression session. Each session was trained with this BioBERT model as a base model with a label that we generated and other considerations. And we had, separate model 1 and model 2. And then we ensemble it. Ensemble, it simply means summarize, and depending on the objective, the way of summarization can be different. So we summarize it to get a final model, and we get the testing data into the final model to tell us how well this model performs. So there's a lot of challenges in the process of, as I have, you may have mentioned, the model needs to be trained on enough reports that actually have BM in it, have a BM yes reports. However, the BM, as we know, it is a rare event, and we don't really know the post-diagnosis BM event rate. The brain head imaging, they can be performed for many reasons other than suspicion of BM. And we have 300,000 reports. It's not possible to label all of them. Unless they are labeled, they cannot be used for training. So we have this issue we need to deal with. So we come up with a 3-stage iterative model training evaluation process. So first stage, we utilized the Alberta Cancer Registry's diagnosis information to enrich the BM yes reports in the training sample. So based on the diagnosis, at cancer diagnosis, we know who are the patients had BM at their cancer diagnosis, and who are patients had no brain tumor, basically no primary or secondary brain tumor, and who had a primary brain tumor, but they are malignant, who are primary brain tumor, who are non-malignant. And it's important for the algorithm to have the ability to differentiate all these types. And these are a summary of what the data we have. And we sampled 868 patients with 868 reports from this patient. As you can see, majority of them are no BM and no PBT, and we had to take a small sample of that. Otherwise, if we just take a random sample, it will be dominated by the no BM and no PBT patients, and we don't have enough BM labels to train our data. Okay. So for the second step, we trained our model, right? We trained our model on the stage 1 training data. And we use that model to predict the BM labels, the BM probabilities on a random sample of 30,000 radiology reports. So now its model predicted the probability. Because it's a model predicted probability, it has a range. It's from 0 to 1. It can take any value. It can say 0.2, it can say 0.5. So we kind of bin them. We put them into, depending on what are the probabilities, we put them into these probability bins. And this is the distribution of these reports in these probability bins, and most of them, a majority of them, are classified as basically had a low likelihood of a BM reports containing BM diagnosis, and we sampled the reports, according to the bins, and these are the samples we take, and for the sample reports, we then have our annotator to label it to get the gold standard label, yes or no, for brain mets, and then we train our model second round. And lastly, we evaluate the data. We evaluate data using post-diagnosis radiology reports from a cohort of cancer patients who are at risk of developing BM. And those are the patients specifically, they're diagnosed during this 1-year time period with follow-up to 2022. So it's like almost 10-year, 9-year follow-up time. They were sampled based on the number of reports per patient. Because first, there's a lot of patients here. A lot of patients don't have reports. Okay, so there's 0 reports people. It's not included here, but there are patients with 1 report, 2 report, up to like 30, 40 reports per patient. So we segmented them into different stratum based on the number of reports they have during their follow-up years. And we sampled by patient. So once the patient is selected from this each bin, we will annotate all the report associated with that patient. Okay, and we annotate them, and we blind the algorithm to this label, and then we asked the algorithm to predict the label, and this way we can evaluate them how well the model performed. And the next slide gives you the model performance. So BMES is when at least one of the model. Remember, we have 2 models, right? One built on impression session, one built on the finding session. At least one of the model predict the probability is greater than a threshold. Okay, and we can choose the threshold to be different values. And the BM no is when predicted probability from both models is less than the threshold. And then there are different measures that measure the model performance. I'm going to just focus on the 3 of them: precision, sensitivity, and the test accuracy. Precision essentially means the proportion of true BM yes report in all the report that are our model said they are BM yes. But our model may be wrong, right? So we have to find out how many of them are actually a BMES by the annotators, the classification. And then this precision is also known as a positive predictive value. And then the other important measure is sensitivity. Essentially, it refers to the proportion of true BMES being predicted as BMES. So of all the actual BMES report, how many of them is predicted by our algorithm to be BMES? It is also called a recall. So we want both precision and sensitivity to be high. And in this case, the threshold, we use different threshold. The different threshold will give you a kind of trade-off between the precision and the sensitivity. But we find this 0.9 recall rate or sensitivity rate to be workable. Basically 90%, close to 90% of the BMES report was identified by our algorithm to be a BMES report. Okay, and the last one is test accuracy. Accuracy basically is the proportion of correct prediction. So the model makes a correct prediction. They predict yes as yes, no as no. And that number is 0.94. It's like 95%, it's close to 95%, which is very, very well. So remember our goal, we developed this algorithm, but our goal is really to understand the BM incidence. The knowledge gap is on the lack of information on when, how often, and in whom the BM occur, especially after their cancer diagnosis. Because at the cancer diagnosis, the cancer registry is doing a good job of capturing them, but not after the post-cancer diagnosis. So we are using our algorithm as we developed to do this, to investigate that, to give us an estimate of the BM incidence. For these, we have study populations that are patients who are at risk for post-diagnosis brain mets. So first, they're not diagnosed with non-melanoma skin cancer. They're not already diagnosed with a primary CNS tumor, and they don't have a BM at diagnosis. So if you already have BM at diagnosis, it's already been captured. Those are not the population we are interested in. All right, so this is a kind of it's a view of the entire study, but I'm going to focus only on the red colored how do I say in the box, the red box, black box colored ones. So these are all the patients diagnosed between this time period in Alberta, 1400. And 1400. Sorry, I think I probably get these numbers, but the number right, whatever I written here is correct. Okay, and after the excluding those, apply this exclusion criteria we just discussed, we have this cohort of 100,000, about 100,000 cancer patient. Out of these 100 cancer patient, 7,000 of them, 70,000 of them, they did not have any radiology reports of their head or their brain. So they were deemed they never had post-diagnosis BM. And the other 34,000 cancer patients, they did have follow up. They did have radiology reports. So those are the patients who possibly would have the post-diagnosis BM. And what we did is we used the NLP algorithm I just mentioned to predict on their radiology reports whether or not that report is a yes or no. And then we summarize all this information to provide this table here. So the BM yes and no here refer to our algorithm predicted yes or no label. Okay, so this is basically the yes means we have 3,299 patients. Out of these 100,000 patients predicted to have a BM post-diagnosis. And the follow-up years for this cohort is 3.8 years median, and it had between 1.8 to 6.4 because we have a long diagnosed frame. And we looked at how it is distributed according to sex, age, cancer site. That's the original cancer. What are their original cancers? And what this is the cancers, the stage at the cancer diagnosis, what the cancer stage are. And these are the numbers. We looked at we compared the no and yes group, and we found all these all these are statistically significant, basically, marginally, whether or not you have a BM diagnosis is associated with sex, with age, with which cancer, original cancer site, and what kind of stage you are diagnosed, the patient is diagnosed in. Okay, so this is it's a lot of information, very rich, so I'm not going to spend more time here. I think another important interest of us is to understand the cumulative incidence rate. So this is the time on the x-axis is time since cancer diagnosis. On the y-axis is the cumulative incidence rate. So at cancer diagnosis, this is 1.64% of patients have BM. This is at diagnosis rate. And we added the 10-year over time, how the BM is diagnosed over time. And at 10 years, we got this number estimate, which is 6.74. This is point estimate, and the red color, you see the shade represent a confidence interval of this estimate. And based on this median of 3.8 years follow-up, we also calculated the incident rate of BM is to be 700 to 23.5 per 100,000 person year. So some discussion and the future work, we had developed this algorithm. Now we are going to do the algorithm validation in Ontario and the British Columbia with our collaborators. And if everything goes well, we are going to hopefully to see to the implementations of our algorithm into the workflow in the cancer registry. So hopefully then the cancer registry will be able to capture this important information into the registry database. And the other thing is we find the cumulative incidence of BM increase more rapidly during the first few years following diagnosis. Then it seems to be flat a little bit over time. This is based on the observation from the curve. I'd like to acknowledge our project team, our core project team in Alberta, Zorina, Valerie, Dr. Lili Mo, that's myself, and Han Xiao. The three are students. Dr. Mo and me are the co-PI for the project. And we have a core team in Ontario, Dr. Darth, with his team at the St. Michael's Hospital. And in British Columbia, the core team involves Jonathan and Lovedeep at the cancer registry there. And we appreciate our annotators and collaborators. Annotators are Eugene, Marco, Ali. Our collaborators in Alberta, we have Jay and Derek, Angela, Emily, Lorraine, Faith. In the BC, we have Stephen and Ellen, and in Ontario, we have Arjun and Mary Jane King. And of course, we are grateful for all the funding agencies. Canadian Cancer Registry had supported this research. Our Brain Tumor Foundation of Canada has been a constant support for our research, and Alberta Health Services provided enormous facility and resources to us so we can carry on this research. Lastly, I want to just say one more word. Stay tuned for the webinar to come in the fall 2025, and where we can give a close look at BTR SRC and our work on the primary CNS tumor surveillance. Okay, that's all. Thank you very much for your time.
Thank you so much, Dr. One. That was great. I certainly learned a lot. And I'm going to encourage folks to add your questions into the Q&A or the chat if that is easier for you. But seeing lots of positive feedback, which is great. I can start us off with a question. Something I'm curious about. So when you were describing that knowledge gap, you said there's that lack of info on when and in whom and how often BM occurs. And then you looked at all those variables, so you have lots of rich data. But I'm curious if there's any other variables you might be interested in introducing to the model that could help you learn more about that knowledge gap.
I think the important, not the important, but important clinical knowledge is, who are those patients and at what kind of rate they are appearing and when they're appearing, right? Are they appearing the first 2 years, or they are, as we see in that curve, it's kind of still increasing at 10 years, still increasing. So definitely those are, you know, we need to we cannot discount the importance of the follow-up, and also without this detailed information on like, okay, so it's, I shared a part of my our results. The other part is on the 1 year diagnosis group. On this 1 year diagnosis group, we do have 9 years of follow-up data. And when we compare this 9 years diagnosis group's data with the shorter time frame of data, we can see actually which cancer site had more brain mets at a later time. We can do that. We can find out that information. I think that are all very interesting and important informations, and I think it's important to work with the clinicians to find out what are important clinical questions and important variables uh, we should be taking into account. I guess, for example, it would be helpful to know what kind of treatment patients went under, and whether or not, the cancer treatment has anything to do with their preventing, right? Whether or not they can prevent the pre-mets, that would be important information to know. But at the moment, we don't have that level of details, but that's something we can totally work towards on once we have the data.
Yeah, absolutely. Sounds like there's a lot of potential to grow, which is great. There is a question. Is there any information on the rate of diagnosis? Is it now 1 in 27?
Sorry, I don't understand the 1 in 27. What is 1 in 27?
I I believe 1 in 27 folks who are diagnosed also with brain mets is my guess.
1 in 27 also diagnosed with the brain mets. Is that, do you refer to the cumulative incidence rate? I don't see the 1 in 27, sorry, in my own
Oh,
slides, so I cannot translate.
apologies. Yeah, so I just got additional information. 1 in 27 Canadians are diagnosed with a brain tumor daily. So is that reflective of your data as well?
Oh, this refers to about the incidence. I think in early slides, we said we estimated there's about 10,000 brain tumors being diagnosed annually in Canada. So you can divide the 10,000 by 365 and get the number about is that, 27 tumors, and whether or not if it's 27, then it include a primary brain tumor and secondary brain tumor. But if it's small, then it means it's only primary brain tumor we're talking about. Yeah, I have to do the calculation, sorry.
Yes, of course, not a problem. There's another question. Is there a connection or study between brain tumors and autoimmune conditions like MS?
Sorry, that's beyond my knowledge. Yeah, I can certainly look into that. Literature review.
Yeah, not a problem. Not seeing any other questions. We'll wait just another moment here.
Mhm.
Okay, there is one other question. Have you tried using your AI program to predict reoccurrence rate for various types of brain tumors? For example, we have been told that oligodendroglioma, sorry, hard one to pronounce, has a high risk of recurring. Does your research show that?
Oh, that is definitely something we will be interested to do. This, as I explained, this is the first time I ever present this work. We only got the algorithm developed, and we only started to, first we're going to validate it in other provinces to say whether or not this algorithm is generalizable, right? Can be useful to other jurisdictions as well. Then we can go into more other arenas. For example, like like your audience asked the question, the recurrence of some uh, other tumors. Because right now, this particular one algorithm I'm talking about, it only detect brain mets. Yeah, for different recurrence, it's possibly we need to develop slightly different algorithm. We
Yeah,
need modifications, essentially. We need to test how well, for example, whether or not this algorithm can be transported to do the same task, right, for primary brain tumor recurrence.
absolutely. Thank you.
Mhm.
And I'm seeing one last question here. Just asking about the timeline of your projects. This person is curious about how long you've been using AI in your work or in healthcare research in general.
Well, that actually also depend on what's your definition of AI. I'm trained as a biostatistician, so I'm very much I used a lot of machine learning algorithms during my doing my own research, starting from my graduate program. So I've been using machine learning algorithms, which you can classify them as AI, but you can also classify them as something like a uh, 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 preliminary AI, like a pseudo AI, or like a less of AI, but more AI like than anything else. I don't know. I don't know how to describe it. I've been using that for a very long time. But this particular NLP project and working with text data, this is my first time doing this type of project, which I started, as I said, started 2021 or 2022-ish. Yeah.
Wonderful. Thank you so much, Dr. Wan.